Hello, I'm Paul Luchenko, and in this sixth video, I'll talk about evidence which suggests that the head direction cell system is essential for telling locations apart. The story begins as follows. We know from this elegant study from the lab of Edvard and May-Britt Moser that head direction cells synapse directly on place cells in the hippocampus. So our question is as follows. If place cells are sensitive to the direction of a maze compartment, are they getting this directional information from head direction cells? Our way of answering this question is as follows. If we get rid of the head direction cells, what would happen to the place cells in our maze with compartments facing different directions? What we'd predict is that the place cells would no longer be sensitive to direction and would thus fire in the same way in each of the 4 compartments. So this is what we did. We wanted to get rid of the head direction cell input to the place cells to see if the place cells would no longer be able to tell compartments facing different directions apart. Now, the anatomy is slightly complicated here. Though the proximal connection between the head direction cells and the place cells occurs in the medial entorhinal cortex projections to the hippocampus, medial entorhinal cortex being MEC, the source of the head direction cell signal is further upstream, at least according to the evidence from the laboratories of Pat Sharp and Jeff Taube. Thus, to remove the entire circuit, or at least the majority of the circuit as we know it today, we can get rid of the lateral mammillary nucleus. Doing so should block the head direction cell information from getting to the hippocampus. So this is preliminary data from control animals, that is rats who still have their head direction cell system intact. What we find in these animals is that place cells show repeated fields in compartments that face the same direction, but only show a single field when the compartments face different directions. This again suggests that place cells are sensitive to direction. These recordings, I should say, were done by Dr. Bruce Harlan and Dr. Roddy Greaves. So what happens in animals without a head direction cell system? As in the control animals, rats with a removal of the lateral mammillary nuclei, the potential source of head direction cell signals, show repeated fields when the compartments face the same direction. In this way, they are similar to the place cells we saw in our control animals. What we observed in the same compartments facing different directions, however, was striking. Here, place cells tended to fire in most or all of the compartments. So to summarize, as we had seen before, in normal rats or control animals, we see that place cells show repeated fields when we have compartments that are facing the same direction. And they show less repetition when the same compartments are arranged in different directions. However, when we removed the head direction cell system by removing or lesioning the lateral mammillary nuclei, we saw that the place cells also showed repetition when the compartments or the rooms faced different directions. So what that suggests to us is that head direction cells may work closely with place cells and allow them to be able to distinguish environments which look similar but which face different directions.